Hello everybody, good day. So we will continue the regulation of gluconeogenesis. Then we will enter into the Cori cycle. Okay, before we close the lecture of gluconeogenesis and glycogen. Okay, like I told you before, remember the three regulated points of glycolysis. All of these points in glycolysis is the regulated points for gluconeogenesis as well. But because one of the points needs to reaction to bypass or to reverse the reaction of glycolysis, in gluconeogenesis, the regulated points altogether becomes 4. Okay, so you can take a look at the type of the activator and also the type of inhibitor of each reaction. For example, at reaction number 3, okay, the regulated point of glycolysis, the inhibitor could be citrate and ATP because that signals a lot of energy. So it tells the body do not break the glucose anymore because we already have a lot of energy. But the presence of AMP and also certain hormones such as fructose 2,6 biphosphate signals that the body doesn't have enough energy, so more glucose needs to be broken down. So everything that can be activated here, glycolysis, will become inhibitor, okay, in the reverse reaction, gluconeogenesis. For example, AMP is activator here, but here in gluconeogenesis, AMP will become inhibitor. Okay, because it shows that the body doesn't have enough energy, so the liver needs to work extra hard to produce the glucose to supply the glucose to other cells in the body. Okay, the same for this one as well. And this one is ester CoA because this ester CoA usually being produced during the fat metabolism. Okay, remember the fatty acid is a very long molecule and it will cut 2 carbon each from the fatty acid to form ester CoA. So if there is a lot of ester CoA in the liver, what will happen is ester CoA can act as a substrate for the formation of glucose, so that glucose can be sent to other cells in the body. Okay, so this is also the summary of the gluconeogenesis, glycolysis, and also glycogen formation. As you can see from here, it starts from glucose, and in the muscle cell, okay, glucose can be broken down through glycolysis and finally form the pyruvate through the action of the first enzyme hexokinase. Okay, this is one of the regulated points in the muscle cell. Okay, and in the reverse reaction, as you can see from here, pyruvate needs to produce oxalic acetate first. We will see a lot of oxalic acetate later on during the CA cycle, and eventually it can produce the glucose again. Okay, by the reverse reaction, except for or unless for the reaction at reaction number 1, 3, and 10. As you can see from here, glucose 6-phosphate need to convert it to glucose, so it will use glucose 6-phosphatase to cut this one phosphate from glucose and free the glucose from the cells. Okay, so the main function of phosphate here is actually to trap the glucose inside the cell. And we know that in the liver and in the muscle cell as well, I think also in the adipose tissue, if we have extra glucose, the reaction will be slowed down, okay, at reaction number 1, 3, and 10. And instead of producing pyruvate, it will be redirected to produce the glycogen. So the formation of glycogen is through glycogen synthase. But if the muscle doesn't have enough energy, okay, this one is in muscle, glycogen will be broken down through glycogen phosphorylase by adding 1 phosphate. And this glycogen will produce glucose 1-phosphate when it cuts through the glycogen phosphorylase. Remember the linear one, okay, the alpha 1,6 glucosidase bond, alpha 1,6 glycosidic bond, it will produce glucose 1-phosphate. So that's why this one needs to go isomerase first to produce glucose 6-phosphate. And we know that glucose to glucose 6-phosphate requires 1 ATP, but this one doesn't have ATP. So instead of 2 net ATP, the ATP that we got from glycogen up to pyruvate just will net the total of 3 ATP, extra 1 ATP compared to the glycolysis. Alright, this is a summary of everything. Okay, now we will enter into the Cori cycle. One of you guys asked me before about how the body recycle the use of lactic acid. So the Cori cycle operates during exercise when we don't have enough oxygen. That's why it will be converted to lactic acid when aerobic metabolism in muscle cannot keep up with energy needs. Glucose synthesized in liver and transported to muscle and the blood. Okay, because when we exercise, we use a lot of glucose. So the glucose inside the blood vessel will be low. Okay, and the liver will compensate this one by converting the glycogen or synthesize the glucose through gluconeogenesis. A highly exercising muscle generates a lot of NADH from glycolysis. Okay, for this one, we will take a look at this figure. Okay, so in the muscle, also in the red blood cell, I told you before, red blood cell doesn't have any organelle, so they always perform the anaerobic respiration. Okay, and others that requires that will convert to lactic acid under the absence of oxygen. So glucose will undergo glycolysis and produce 2-pyruvate. In the process, it will require 2 coenzyme, 2 NAD plus to becomes 2 NADH and produce net 2 ATP. But the problem now, although we have 2 ATP and 2 pyruvate, we will have a lot of NADH. This NADH must be converted back to NAD plus. So the next glycolysis can occur because the amount of NAD plus is limited in the body. So if they are being converted to NADH, they must be converted back to NAD plus. Under normal condition, this process, this conversion back to NAD plus can happen in electron transport chain. But because we don't have enough oxygen, no electron transport chain will happen. Alright. So this 2-pyruvate will be converted to lactic acid, or lactate to convert back this NADH to NAD plus. Now the next problem is we have a lot of lactate in the body. So this lactate will be transported through the blood to the liver, okay? And renal cortex is the kidney. I told you before these two are related. This 2 lactate will be converted back to pyruvate because we don't want lactic acid in the body, right? It's acidic. In the process, it will utilize enzyme lactate dehydrogenase. Okay? Remember, if dehydrogenase is always a redox reaction. When it's redox reaction, it always involves the coenzyme. Okay? So this 2 NAD plus will be converted to NADH. Now we have back the pyruvate, but we have a lot of NADH in the body. It must be converted back to NAD plus. So in order to convert back to NAD plus, this 2-pyruvate will undergo gluconeogenesis and produce glucose. And this glucose can be transported back in the blood, okay, and to all the cells in the body that requires the glucose. So in this process, this NADH, okay, will be converted back to NAD plus. So the cycle of the reaction can continue to happen. Okay, so this is basically the explanation of the earlier figure, right? Okay, now usually in the exam, we are not going to ask you straightforward question, like why Cori cycle needs to happen, okay, because that's going to be too easy. So usually we will ask something like explain why the consumption of alcohol can lead to the disruption in the Cori cycle. Okay, for example, so we will go one by one. So in order for you to understand this one, you need to understand the Cori cycle and gluconeogenesis first. So metabolism of ethanol alters the NAD plus and NADH ratio in the cells. But if you drink alcohol, okay, usually in the form of ethanol or any kind of alcohol, the primary site for ethanol metabolism is liver. This one. During the ethanol metabolism, what happens is this ethanol will be converted to acetate or acetaldehyde by the formation of NADH. So these 2 NAD plus and this NAD plus will be converted to NADH, and this one can be converted to NADH as well. Now our body contains lots of NADH but low NAD plus. So any reaction that requires NAD plus will be disrupted. Okay, for example, gluconeogenesis or the formation of lactic acid. Okay, so because we can see from here, remember, anything that is requiring NADH will be disrupted. Okay, so for example, just now I told you it happens in the liver, right? So it won't kacau here. It won't disrupt the process here, but it will disrupt the process here. So if lactic acid to pyruvate requires NAD+. Okay, now we have lots of NAD dishes but less NAD+. So this lactic acid cannot be converted to pyruvate. Okay, and because of that, the body will contain lots of lactic acid. Okay, and the body will become acidic. Okay, other reaction that requires the NAD+ as well, for example, fatty acid oxidation and breakdown, can also be disrupted. So that's why people who consume a lot of alcohol, their liver becomes fatty because the fatty acid uh, cannot be oxidized or cannot be broken down into smaller portion, okay, to be used as energy, and their body will become acidic because liver cannot convert the lactic acid back to the pyruvate. Okay, and on top of that, acetaldehyde or acetate can escape from the liver and affect, maybe it can disrupt some of the cells in the body and cause the cells to be killed or oxidized, and also some of it can cause drunkenness, okay, because it can enter into the brain. Okay, so there are many bad effects because of the alcoholic consumption, but remember, everything, because in food, everything has their own dose of poison. So if you guys are drinking alcohol, especially for the non-Muslim, it's okay, but at certain amount. Okay? Because our body will always have a mechanism to balance the anything that we eat, anything that we put into our body. So probably you can drink 2 or 3 two to three cans of alcohol, but not more than that. Because the escaping of acetaldehyde, or some of the Asian people especially, they can metabolize the acetaldehyde very well. Your face sometimes can become flush, okay, red. So that's why people who drink a lot of alcohol, sometimes your face is red. Okay, you can read these consequences yourself. I already explained to you before. For example, formation of the fatty liver, inhibition of gluconeogenesis in the liver. This one all happens in the liver, not yet in other cell. Remember, so acetaldehyde can escape into the liver. Okay? So it can cause hangover and so on, because it's very reactive. Okay. So this is types of energy utilization. This one is not very important, but I just tell you a little bit. So for the anaerobic energy system, usually it is utilized when you need the explosive energy. Okay? For example, when you are weightlifting, so you will need explosive energy. So you will undergo the anaerobic respiration. You will produce very fast energy, net to ATP, but very inefficient. Certain microorganisms, they prefer anaerobic respiration, of course. And for example, fish, certain type of fish in the sea, they will uh, perform the anaerobic respiration because they requires an explosive energy during what we call it, swimming. Okay, and then they will need to stop and swim slowly because then they will have the oxygen depth. Okay, and they will do the aerobic respiration during rest. So aerobic respiration we produce a much better energy percentage, or much more efficient, but they are slower. Okay, so people who are doing long distance running, okay, who can pay back their oxygen debt during the slow pace of the exercise, they all will use aerobic respiration system. And usually, if you are good enough in exercising, you will start to utilize both of them, aerobic and anaerobic respiration system. So at the start, you will use aerobic, and then you will turn to anaerobic as it can get enough oxygen, and then you will start to deplete your oxygen. That's why sometimes you see the long distance run, they start to consume glucose in the middle of the running, okay, because the glycogen already depleted. So when the glycogen stores has been used up, the muscle tend to cover the energy needs from the fat metabolism. Okay, so fat metabolism is slower, but they can produce a lot more energy compared to the normal carbohydrate metabolism. Okay, so this concludes our lecture for the gluconeogenesis and glycogen as well. Okay, next we will enter into the TCA cycle and electron transport chain. As usual, always come to see me or send me an email or just text me if you have any question that you want. Okay, better if you can type your question in the chat box during our Zoom meeting. Thank you.